Thank you, David. Good morning, ladies and gentlemen. First, I'd like to begin by thanking the forum for the opportunity to speak today. It's a great honor for me, and I really want to use my time well. So thank you. And I begin my talk with an apology, and that is that I have to chair a meeting at the NIH at 10 o'clock. So I'm going to give my talk, we'll have questions, then I have to leave, and then I'll come back in the late afternoon after the meeting. And I'm very sorry for this, and I'm really sorry for the terrific talks I'm going to miss, but I hope to be here for the rest of the meeting. So thank you. So as David said, I'd like to begin this meeting with kind of an overview about how the organisms that live in humans lead to health and disease. And I'm going to take some examples from our own work and from other people's work to try to weave a story. So I'd like to begin with this summary that was published about almost 10 years ago by Larry Colonel from Hawaii, looking at cancer risks in Japanese immigrants to the United States. And there are 4 different cancers that are looked at, and there are also 4 different colors. So they compared, and this is a compilation of prior studies, they compared cancer risk in Japanese people in Japan, first-generation migrants to Hawaii, second-generation migrants, and they compared their risks with Caucasians in Hawaii. And the results are shown here. For stomach cancer, the Japanese in Japan had high rates, the Caucasians low, and with each generation, the rates went down. And we now know that there's a microbe involved in this. But it is interesting to think about these other cancers. Breast cancer, which is changing in the opposite direction in a stepwise manner. Prostate cancer may be the same. Colon cancer looks like one step. We don't know what the organisms are here. We don't even know if they're organisms. But it is interesting to see how the world is changing. Some ways good, some ways bad. So let's begin at the beginning and remind you that animals have been colonized by microbes ever since animals appeared on the planet about 500 million years ago. These microbes are ancient. They live in particular niches in their animal host. Many of them are persistent for the lifetime of their host. Some of them are conserved. For example, there are certain microbes that are present in everyone in this room, and then there are others that are host-specific, where only one person in this room has a particular microbe. I have a couple of favorite slides. This is one of them. This is a microbiologist's view of the human body. Human cells, 10 to the 13th. Microbial cells, 10 to the 14th. Who are they? We know a lot more about who they are as a result of the Human Microbiome Project, which was a constellation of hundreds of investigators whose work is appearing in scientific journals in the last few years and continuing to appear. This slide is from one of the major studies of the HMP, looking at the relationship between where the microbes live and their diversity. This is a principal coordinates analysis looking at microbial communities in about 250 people. The communities are color-coded by anatomical site, and each circle represents one community and one person. So the first thing you can see is that the communities aren't random. They're not just all over the map, but they're clustered, and they're clustered by the site of where the community exists. And yet there's a substantial diversity within the community between people. And that study concerned people in the United States. And I want to point out that we have a very U.S.-centric view, but it's not, that shouldn't be the only view. And this is from studies that we've done with Rob Knight and his group and Maria Gloria Dominguez, comparing the cutaneous microbiome of the forearm in people in the United States and people, Amerindians in Venezuela. And here's Boulder in New York, and here are the Amerindian communities, and there's essentially no overlap. And in fact, here we find 2 different types of communities that don't exist in the U.S. So it's a big world out there, and we're going to have to explore that diversity. Now, a very important study was published early this summer by Jeff Gordon and his colleagues, Tanya Yatsenenko and Rob Knight and Maria Gloria Dominguez, and they asked a number of different questions. I want to highlight this graph from their work, where they asked, when does the adult gut microbiome become established? And they looked at people in the U.S., but also Amerindians in Venezuela and Malawians in Africa. And on this axis is age, and on this axis is the distance to adulthood, in essence. And what they found is that in older children, their microbiota is very adult. There's very low distance. But in young children, the distance is far, and then gradually the children become more and more adult-like. And what's important that I took from their study is this happens by the age of 3. So this is really the critical period in which the developing microbiota is dynamic. And, of course, this is the period of time when children are developing as well. So we can take the geneticist view of the human body and say that there are 23,000 human genes and there are more than 8 million microbial genes. So really, the interaction, just numerically, these guys are favored, but as we know, it's a complex interaction. So we can ask a series of biological questions. What's the identity of the microbes that populate their host? What are they doing? How is the host responding to them? What are the forces that maintain equilibrium among the population? We've been particularly interested in this. What are the unique characteristics of each individual? I want to focus for a moment on a very interesting study done by Ruth Lay and her colleagues, Omri Koren et al., that was published last year, where she looked at the microbiota of pregnant women. And in this principal coordinates analysis, she's comparing the microbiota in the first and third trimester, and this is fecal microbiota, with people with data from the HMP, from males and females. So if you look at the fecal microbiota of HMP males and females, they largely overlap. There doesn't appear to be any strong signal. And first trimester microbiota is clearly different. So during pregnancy, the microbiota is changing, and it's not changing in a random direction, it's changing in a particular direction. And then by the third trimester, it's changing even more in a different direction. So there's a marked evolution of the microbiota during pregnancy, and with perhaps more individual variation. This might be what the moms are giving their babies. So Ruth and her colleagues did an experiment where they transferred the microbiota from the first trimester feces or the third trimester species to germ-free mice. And some of the interesting results are shown here. They found that the third trimester inoculum caused higher levels of cytokines in the CagA of the germ-free recipients, now conventionalized. But very importantly, they found that there was increased adiposity in the recipient, and there was more glucose intolerance. And this is interesting because we all know that pregnant women gain weight and they become more glucose intolerant. And this work suggests that there's a role of the microbiota in moving in that direction, and one can even think of it in an evolutionary sense that the microbiota is helping the partition of energy from one generation to the next. So on the basis of these and many other kinds of observations, we've developed a schematic of the interaction between co-evolved colonizing microbes and their host, in which coevolution could be defined as microbes that know how to talk to the host and to receive signals back, to engage in a very lively conversation. And our view is that this is a dynamic equilibrium. We began it with the concept of a single organism, but have since expanded it to communities of organisms. Work with Denise Kirshner has shown that such a system is robust and resilient. What we're particularly interested is what happens when this gets perturbed. So to consider the perturbation, I'd like to talk about the organism that David mentioned, Helicobacter pylori, And here's Helicobacter in the stomach of a human volunteer. It's a spiral gram-negative organism. Helicobacter was discovered as a pathogen associated with peptic ulcer disease. But we now know from genetic imprints that it's been present in humans for at least 100,000 years, probably longer, present everywhere in the world, acquired early in life and persisting until the death, and highly prevalent in just about in most countries of the world until recently. Now, Helicobacter is a very smart organism, and I'm just going to show you one example of Helicobacter's brilliance. Helicobacter produces a protein called CagA, which it then injects into human cells where where tyrosine domains on the protein are recognized as self by kinases like SARC and ABL, which phosphorylate it. And now this phosphoprotein has many different interactions in the cells of the epithelium. We're going to come back to the consequences of Helicobacter having this system. Now, despite Helicobacter's brilliance, all is not good for Helicobacter. And this slide is from studies that we did with Yu Chen from NHANES 3 and NHANES 4, looking at Helicobacter prevalence in the United States by the age of individuals and by the year of birth in both males and females. And the data are clear. People born early in the 20th century have Helicobacter, and people born late in the century less often have it. And these are cross-sectional data, but longitudinal and birth cohort data show the same phenomenon. Helicobacter pylori, which has been present in humans for at least 100,000 years, is largely disappearing over the course of a century. So what are the consequences of the disappearance? Because Helicobacter causes a host response in the gastric epithelium, we and others asked the question, could it be related to the risk of gastric cancer? And this slide shows studies from the early 1990s by David Foreman's group, Julie Parsonette, our studies with Abraham Nomura, which all show the same phenomenon. People who have Helicobacter in their stomach are at increased risk for developing gastric cancer. And if you adjust for confounders, the odds ratio is somewhere around 9. There's a range around that going up to 40. Helicobacter is the leading risk factor for gastric cancer in the world, and that's now been very clear. So this is clearly a biological cost to having the organism. So I told you that Helicobacter is disappearing, and so is gastric cancer, which is not surprising. In 1900, gastric cancer was the leading cause of cancer death in the United States, and as you can see, it's gone down dramatically and in other developed countries of the world. But at the same time, other cancers have risen. And this slide looks at a series of other cancers, and in particular, I want to highlight adenocarcinoma of the esophagus, which has risen six-fold over the course of about 25 years. It's the fastest rising cancer in the United States and in other developed countries. And for a number of reasons, we were interested in the question is, if this is going up while Helicobacter is going down, is there any relationship? So we conducted the same kind of studies that we did with gastric cancer, but now looking at esophageal adenocarcinoma and GE junction cancers. And this is from the work we did with Wang Ho Chow and Bill Blott and Joseph Fermini at the National Cancer Institute, looking at patients in SEER. And the data are organized so that people who don't have Helicobacter, the reference group, odds ratio 1. If they have a CAG negative strain, odds ratio 1.1, not much different. CAG positive, odds ratio 0.4, and inverse association with Helicobacter pylori, consistent with the idea that Helicobacter is protecting against this cancer. There have been many other studies since then that have shown the same phenomenon. And I want to point to a recent study looking at precursor lesion to adenocarcinoma of the esophagus, Barrett's esophagus, from Doug Corley and Julie Parsonette and colleagues, which show the same kind of phenomenon, same organization, even a stronger inverse association with CAG-positive strains, protection associated with protection against Barrett's. Now, because of the relationship between Helicobacter, the inverse relationship between Helicobacter and esophageal disease, we were interested in asthma. And in part because of the studies by Kurzicki and colleagues that looked at asthma risk at children at the age of 7, according to antibiotic use, and they studied 13,000 kids in Manitoba. The reference group were kids who didn't get any antibiotics. What they showed is that the more courses of antibiotics they received, the higher their risk of asthma. And just because there could be confounding as a result of respiratory infection here, they looked at non-respiratory infection, and the odds ratio was in fact even stronger. So we asked the question, looking at the NHANES database, what's the relationship between asthma and Helicobacter? Well, actually, before that, we did the study in New York with Joan Reibman at Bellevue Hospital, looking at the asthma center, looking at 318 asthma cases, 208 controls. They had spirometry and allergy testing, and we blindly assessed Helicobacter and CagA status. And the major results are shown here, again, with the same organization. CAG negative strains, odds ratio 1. No Helicobacter, odds ratio 1. CAG negative, almost 1. CAG positive, inverse association, statistically significant. When we looked at the universe of individuals in this study who ultimately developed asthma, we did this Kaplan-Meier analysis. Everybody started out life asthma-free. Everyone developed asthma by our definition. But when we look at the age of onset of asthma, those people who had Helicobacter, their average age of onset was 21. And then those who didn't have Helicobacter, their average age of onset was 11. This was a highly significant difference. Because of that, we then looked at the NHANES data, and this is work done with Dr. Yu Chen, an epidemiologist at NYU. Same kind of organization of the data. When we looked at adult onset asthma, there was no relationship between that and Helicobacter. But when we looked at childhood onset asthma, we found, again, the inverse relationship, very similar to the result seen in New York. And this is from NHANES 3. In NHANES 4, we found almost the same result as well. 3 large, blinded, independent studies. The NHANES data also had work on hay fever, and the results are quite similar. No association with adult onset hay fever, strong inverse association with childhood onset hay fever, and even the suggestion of something in the CAG negative strains as well. And in NHANES, they did skin tests on more than 2,000 individuals. And here we're looking at the data for 6 different allergens, and having Helicobacter was associated with an inverse association. In 4 of the 6 cases, it was significant, with reactivity to each of the allergens. Now, what could Helicobacter have to do with Russian thistle, you might ask? So, Ann Mueller and colleagues in Zurich have done a very wonderful series of experimental challenges in mice. And I want to point out to you the study that Isabel Arnold did, in which they used a model of experimental asthma in C57 mice, in which they sensitized the mice to an antigen and then challenged them with the antigen later and then gave them methacholine to see if they could bring out wheezing. And in this experimental construct, they looked at mice who were uninfected or who they deliberately infected with Helicobacter pylori neonatally or as an adult. And in this case, we're looking at the antigen is house dust mite antigen. And so here's one of many studies that they showed. I think it's particularly revealing. In the methacholine challenge, when they didn't give the house dust mite antigen to the mice, this is what their change from baseline is, very, very little change from baseline. If they did give the house dust mite antigen, there's a big change, as expected. The mice were sensitized. They did the equivalent of wheezing. When they were given Helicobacter as adults, there's no difference, but when they were given Helicobacter as neonates, it completely ablated the phenomenon. And we can see this in looking at tissues. Here's the dust mite antigen sensitization, the very inflamed airways. Here's the control. Here's the adult, looks like this. Here's the intranatal, closer to the uninfected. And we see this at many different directions. So let me just summarize. There has been a lot of studies now looking at the changes in physiology from the Helicobacter colonized stomach to the Helicobacter-free stomach. The ancient stomach, where Helicobacter is a major member of the microbiota, in which gastric cancer is the major disease, to the postmodern stomach, free of Helicobacter, in which GE junction cancers are rising. We know that physiologically, the lack of Helicobacter increases somatostatin production, decreases gastrin, increases gastric acidity, has an intermediate effect on leptins. Lack of Helicobacter increases the gastric hormone ghrelin. Helicobacter, the stomach is the major source of ghrelin production. And it also decreases T regulatory cell populations, which are rich in the stomach of individuals who have Helicobacter. There are a series of consequences that have been linked to this. Good news, gastric cancer going down, peptic ulcer disease going down, GERD and its subsequent cancers going up, childhood asthma going up, and questions about obesity, celiac disease, and stroke. There's evidence that Helicobacter, in fact, may help protect against tuberculosis. On the basis of these observations with Helicobacter, we developed something which we've called the disappearing microbiota hypothesis, that changing human ecology beginning in the late 19th century has dramatically altered the transmission and maintenance of our indigenous microbiota. These changes have affected the composition of our microbiota. I point to Helicobacter as an example. The altered composition affects human physiology and thus disease risk. Point again to Helicobacter. And the loss of ancestral bacteria that usually are acquired early in life is especially important because it affects the developmentally critical stage. And I'm going to be talking about that for the rest of my talk, or almost the rest of my talk. So if we go back to the work of Yatsenenko and colleagues who look at the question of when the diversity of the microbiome arises, adults have a certain range of total diversity. But when they summarized it by origin of the adults, they found that there were a lot more OTUs in the people from Africa and the Amerindians from Venezuela than people in the U.S. In fact, the people in the U.S. on average had 200 to 400 fewer OTUs in their fecal specimens. These were statistically significant, supportive of the hypothesis of disappearing microbiota. A few years ago, Stan Falco and I proposed that maternal status is very important for the composition of the residential microbiota of the next generation. And our view was that mothers start life with a certain microbiota, and if they happen to lose those microbiota, unless they have a way to regain them, the next generation starts out poorer, and this continues for generations. And we speculate that this, in fact, is what's been going on during the 20th century. So, in particular, how do mothers transmit their microbiota to children? It begins at birth, through passage through the birth canal, where the baby is coated with and drinks the bacterial, the vaginal microbiota, and then the baby is placed in contact with the mother's skin to the mother's breast. The mother is kissing and licking the baby. There are many ways that the maternal microbiota may be transferred to the child. But we're changing mothers in a lot of ways, like antisepsis, antibiotics, and diet. And the babies are under a lot of different changes as well, such as cesarean sections, which change the founding microbiota of the host, and bathing, and bottle feeding, and of course early life antibiotics, which I'll be talking about extensively. So antibiotics, we credit Fleming's observations on penicillin, published in 1929 at the beginning of the antibiotic era. They really achieved, they began to be widely used in the early 1940s, first in the military and then in the civilian population. Here in 1945, we have a report in the Journal of Clinical Investigation of the treatment of LOBAR pneumonia with penicillin, treatment of 64 patients with remarkable results. Now let's fast forward 65 years. Here we're looking at the top 8 prescriptions in U.S. children, 2010. Of the top 8, 5 of them are antibiotics. And if you summate all these courses of antibiotics, you find that there are 41 million courses of antibiotics given to U.S. children. And if you divide that by the approximately 70 million children under the age of 18, and you do a little math, that would be about 10 courses of antibiotics over the course of 18 years if it were divided equally. If you look at the first 2 years of life, it's more than 1 course of antibiotics per year. And it's not just in the United States. Even in developing countries, antibiotics are widely used. Here in a little bodega out in the Amazon, in between drinks and batteries, are antibiotics for sale over the counter. Antibiotics are everywhere. So we've been particularly interested in one of the rising epidemics, which is obesity. And this slide is from CDC data looking at obesity trends in U.S. adults, 1989 to 2010. In 1989, no state has more than 14% of adults who are obese. By 2010, no state has less than 20% of adults who are obese, and the national average is about 30%. What's important in this slide is first that the changes are happening all over the country, and in fact all over the world. And the other important thing is that this happened in 21 years. Something really dramatic has occurred in those 21 years. So we've been interested in understanding this and have been guided by observations on the farm, where since the 1940s, farmers have been feeding sub-therapeutic levels of antibiotics as growth promoters to their farm animals. The farmers have learned that sub-therapeutic antibiotics increase growth in everywhere from chickens to cows, a very wide swath of vertebrate evolution. Many different antibiotics and other antibacterials work, regardless of their chemistry, their class, their target, or their spectrum. Antifungals and antivirals don't work. The farmers found that the younger they start antibiotics, in this case with swine, comparing starters and grower finishers, the younger they start it, the more profound the effect. And this immediately tells us that it's developmental. So we began to model these phenomena in mice, initially beginning with C57 mice, where we exposed them to subtherapeutic or stat antibiotics or control. The initial work was begun by Il-Sung Cho when he was a fellow in GI at NYU. Il-Sung compared controls in 4 different antibiotic regimens. He didn't find any difference initially in total body mass, but he did find differences in fat, and it's shown here that each of the antibiotic regimens increased the amount of fat in the mice. In this experiment, no effect on lean. Il-Sung's experiments were continued by Lori Cox, a graduate student in the lab. Il-Sung and Shingo Yamanishi, a postdoc from Japan, looked at gene expression in the liver, comparing control and stat, and we're looking at a couple of categories of genes: fatty acid metabolism, lipid metabolic processes. Red is upregulated, blue is downregulated. You can see a huge upregulation of stat, of many of these genes of central metabolism. It's been corroborated by qPCRs as well. The liver is receiving more short-chain fatty acids from the portal circulation and is converting them into fat, which is going out into the periphery. We proposed a series of mechanisms for stat-induced adiposity in animals that they're developing, that antibiotics are mediating differential selection of the colonic microbiota. This is changing composition. It's altering representation of short-chain fatty acid synthesis genes, increasing short-chain fatty acid synthesis, inducing hepatic lipogenic genes, and bringing fat into the periphery. To understand this better, Lori asked the question, can the growth phenotype be transferred by the microbiota? She did cecal transplants from the median of the control group or the stat group and transferred the microbiota to germ-free Swiss Webster mice. Now, those are based on studies on the farm with continuous low dose of antibiotics, but human children are getting high doses of antibiotics to treat their ear infections and their throat infections. So we developed a second model, which we call PAT, high doses of antibiotics. This was done by Yael Noble, a student. Her hypothesis was that a series of short therapeutic dose pulses of antibiotics administered early in life will sufficiently change the gut microbiome to alter body composition. The mice either received no antibiotics or 3 pulses of amoxicillin, the most commonly used antibiotics in children in the United States, or tylosin, a macrolide. We consider this the Z-Pack for mice, or tylosin and amoxicillin mixture. 3 pulses. First pulse, they're all in the drinking water. First pulse, only the mom's drinking it, but the pups get the antibiotics through the mom's breast milk. They're weaned, and on day 41, we put them on a high-fat diet to bring out the phenotype. So, from ecological theory, Payne and colleagues studying forests looked at the effects of large, infrequent disturbances on community state. If there's a stable ecosystem and you perturb it, it generally recovers. If it's perturbed twice, it doesn't. If it's in an intermediate state and you perturb it, it doesn't recover at all. Another way of looking at what we've just talked about specifically in terms of the early life phenotypes is that there are mesenchymal stem cells or stem-like cells that must decide how many times to go around and what should they become, fat, bone, or muscle. We have evidence that antibiotics are changing each of these. In the past, there's been a focus on diet and calories affecting this phenomenon. We propose that it is the nature of the microbiota and the host reactive cells that are sending signals that affect this decision and this decision. I want to acknowledge the people who've done the work on the STAT and PAT research. I've mentioned many of them as we've gone forward. Il-Sung Cho, Lori Cox, Shingo Yamanishi on STAT, Yael Noble on PAT, Alex Alexienko on informatics, Jerry Zavadil on microarrays, Kathy Mahinda Palacier on metagenomics, and a number of other colleagues. Now, in a couple remaining minutes, I want to sum up and bring up a couple more topics. So I want to apply economic theory to infectious diseases. Nothing is free. Sometimes costs are low. Low cost does not mean free. Low cost may be cumulative. I'm concerned about all the antibiotics that we're giving to kids, and I wonder whether there is collateral damage to human children from antibacterial activities, metabolic and immunologic, as we've shown in mice. We have some early data on neurologic abnormalities as well, and we wonder about reproductive. If we think about collateral damage to adults, it could be metabolic, it could be allergic, it could be neoplastic effects. We've already seen that there's the change in the microbiota from Helicobacter dominated to not has changed the ecology of cancers in the upper GI tract. It could also be passed forward. And of course, changing the microbiota increases susceptibility to resistant organisms and potentially to epidemic infections. I just want to close with one last comment, and that's about estrogen-related cancers. In 2012, 3 of the top 10 cancers in women were estrogen-related, and they account for about 300,000 cases, about 60,000 deaths. I want to bring to everybody's attention this very interesting graph that was published in 2003 by Mary Claire King, looking at breast cancer risk in women who have BRCA mutation carriers by period of birth. We know that women who are BRCA positive have a markedly increased risk of breast cancer. But there has been a secular trend. Women born before 1940 had this curve of breast cancer, and women born after 1940 have this curve. It's been shifted this way to earlier life. At any age, these women are more likely to develop breast cancer. What's caused this? And let's also look at endometrial cancer, which here's India, low rates. Here's North America, highest rates. This is a disease of development. And most of endometrial cancer is type 1, which is estrogen receptor positive. So it turns out that estrogens participate in an enterohepatic circulation. This was first really identified by Dr. Adela Cruz in Finland about 50 years ago, and Sherry Gorbach did a lot of important work about 30 years ago. We now know that estrogens circulate in the blood. They go to the liver where they're conjugated. They come out in the bile as conjugates. They're in the GI tract. And then if they remain conjugated, they're excreted. But if they are deconjugated, then they are reabsorbed. So there is an enterohepatic circulation in which the gut microbiome is the switch determining in or out.
Claudia Platel, a talented physician who worked with me, has coined something which she calls the estrogen hypothesis, that it has three key elements, that there's enzymatic activity, bacterial beta-glucuronidases and other enzymes that deconjugate, that this functionally modulates the enterohepatic circulation of estrogens, and that there's a consequence of lifetime estrogen exposure and composition. So we've seen rising evidences of breast cancer and endometrial cancer with socioeconomic development. This is a series of testable hypotheses related to that. And on that note, I want to thank again all my colleagues who are actively developing work on the microbiome, and I'm happy to hear your questions. Thank you very much.
Thank you, Marty. There are a number of interesting topics worthy of discussion. Are there questions or comments from the forum? Claire?
Great talk, Marty. A couple of questions. You know, it's interesting in terms of what you've observed with H. pylori that there really seems to be a trade-off between increased risk for gastric cancer versus esophageal adenocarcinoma. Do you think that it's possible to get at some of the mechanisms driving this shift in risk so that at some point we could have the best of both worlds?
Yeah. So that's a wonderful question. And there's an apparent paradox there, and that is that the strains of Helicobacter that appear most associated with gastric cancer are those that are CagA positive. And those are also the strains that are the most protective. So against esophageal disease and childhood onset asthma. So, and it's not surprising because those are the strains that have the most gain. They are the most interactive with the host. I think this is the provenance of medical research. We have to understand most people who have Helicobacter never develop gastric cancer. So we have to understand what are the factors that push someone in the gastric cancer area. Most people who don't have Helicobacter don't develop esophageal adenocarcinoma. So there are obviously other risk factors that are involving host responses, cofactors as well. It's a complex biology. Could we ever intervene? One hypothetical solution, and again, I didn't have time to go into this, is that if you put things on a scale, I think the evidence shows that Helicobacter is mostly beneficial early in life and is mostly harmful late in life. So it's like a pleiotropic gene that has those kinds of effects. So the consequence of that is that some years from now, doctors of the future will be giving Helicobacter pylori back to children to replace their ancient microbiota that has been lost, and then at some age, 30, 40, 50, eradicated, so as to have the early life benefit and not the late in life cost. And I would suggest that could be a paradigm.
Claire, did you want to follow up?
Yeah, I don't want to hog the floor here, but I have a follow-up question. It really occurred to me when you were showing the slide that was in the paper that you and Stan Falco published about disappearing microbiota over time. One of the questions that came to me, and this is maybe a little bit far out, but I wonder, what are your thoughts on the fact that women are delaying childbirth, and if you posit that the microbiota in a 20-year-old woman may be more diverse than what you see in a 40-year-old woman, are there some direct consequences of that in terms of what's passed on to the child and the subsequent consequences?
Yeah, it's a wonderful question. Again, that's a frontier for study. But my hypothesis is that every time we take an antibiotic, there's some level of extinction. That there's some population, there's some community of organisms that's present at low levels that now is lost. It has oscillated down to zero. And unless there's a way to regain those populations, every time someone takes an antibiotic, there's loss of microbiota. That's a hypothesis.
but it's consistent with the diversity of organisms that we have. And one of the differences between a 20-year-old and a 30-year-old is the 30-year-old has had more courses of antibiotics for their throat infections, and all of the ways that we're using antibiotics as if antibiotics had no cost. I think the most important thing that I want to say is that drugs as powerful as antibiotics must have cost. We've just been ignoring it for the last 60 years.
Marty, I wonder if I could just follow up on a point that Claire raised and something that you touched on. Presumably, or perhaps, within the stomach environment, Helicobacter, although very sort of potent and prodigious in its ability to sort of affect its way in that environment, is doing so in the midst of a more complex community, and you and I know that well. What do we know? What more do we need to know about the other members of the gastric community, which perhaps in a lot of these circumstances where Helicobacter doesn't explain, presence, absence, CagA plus Pygmus doesn't explain the whole story, might there be other biology that we need to know a lot more about that could account for perhaps even a larger fraction of phenotypes either way?
Yeah, a great question, David. The question of competition between Helicobacter and other organisms has not really been explored at all. And I'm very interested in a problem that I've been thinking about for 25 years and still haven't solved. And that is, in women who are Helicobacter, the maternal Helicobacter positivity is the strongest risk factor for a child having Helicobacter pylori. And so the question is, how might Helicobacter get from the mother to the baby? It's never been clarified. And one possibility is that it's happening at birth, through the birth canal, but Helicobacter is just at a very low state, below the radar screen, and then later it blooms, because in populations where the moms are all Helicobacter positive and the babies are destined to become Helicobacter positive, we know this from longitudinal studies, they don't become positive till after the age of 1 or 2. They start turning up positive there. If you look at the signatures of those organisms, they're largely maternal. So somehow the maternal organism has been transmitted to the baby. We don't really know when, but one possibility is that Helicobacter is there, but it's held in check by other organisms, maybe Lactobacillus. And then as Lactobacillus recedes, Helicobacter moves up. That would be a typical ecological succession.
I know you need to leave in a few minutes. How long do we have?
I've got 10 more minutes.
10 more minutes. All right. Would you mind going to a microphone and introducing yourself, if you don't mind? Thanks.
Steal a microphone?
You
can use
it. Please steal.
I'm Michael Zasloff. This is awkward. Very awkward.
Michael, you could go to the front.
More so.
Like sit in front, that might be better.
Here we go.
It's even weirder. Marty, here's my question. The hypothesis that attempts to relate changes in the fecal microbiome to obesity, as you've described it, suggests that the organisms in the rectum or the colon, in some way or another, increase the extraction efficiency to some extent of the food being eaten, or convert it to a greater extent to free fatty acids, which are handled. But in the real world, in an ad lib feeding environment, you'd think that our brains will recognize the increase in energy, and since that's a fundamental feature of human physiology, you'd think that the animal's appetite would drop. So, although the argument goes that, we're getting more nutrients per gram of food, per weight of food, maybe there are other ideas. And the only thing I wanted to mention was there's some very recent work from Richard Gallo in California that demonstrates that if you inject bacteria into the dermis of at least a mouse, you see expansion of adipose tissue. So that's just a thought.
So farmers have found that if they give low doses of antibiotics to their farm animals, the farm animals get bigger. And they've shown 50 years ago that it doesn't work in germ freeze. So it requires the microbiota. And they also have shown that it increases feed efficiency, the conversion of food, measured amounts of food to energy. What are the intermediate steps? That's what we're exploring. It could be increased utilization of food substrates. It could also have to do with host response in different populations of responding cells, like T cells, for example, changing the economy of growth at a critical window of development. And that's, I think, one of the points that I want to make. All this stuff may not help people who are adults and already are obese. It's really about how we might consider trying to prevent further obesity in our children and grandchildren. You mentioned about appetite, and I couldn't but smile because that's what ghrelin does for a living. Ghrelin, when the stomach is empty, ghrelin turns on and says, "I'm hungry, feed me." And Helicobacter is involved in the regulation of ghrelin. We and others have shown that very clearly. So, in fact, it may not just be the colon. It may be the aberrations in the gastric compartment as well, in a more complex model of interaction.
Marty, I think there are two questions if you have time. One is Eduardo and then Richard.
Yes, it's a short question. If you review the data in Venezuela and Malawi, and probably in the United States very early in the 20th century, the quality of water was very poor. That is, the children acquire bacteria very, very early. Maybe this can play a role in the different cancer patterns around the world right now?
Yeah. So I think that the point about water quality is a really excellent point, and something that we're quite interested in. Clean water is desirable. Clean water has saved countless lives because it has decreased the transmission of pathogens. But it probably has also decreased the transmission of commensals. And the question is, what's the balance there? And can we give back the commensals, for example? There's a hypothesis. It may be that chlorination of water, that chlorine has effects on our microbiome. That hasn't been studied yet. So water is important. And until recently, there were a lot of antibiotics in water all over the United States because of runoff from agriculture, where most of the antibiotics used in the United States, even today, are still used, but it's better regulated now. But there's still a lot of antibiotics trace levels of antibiotics in water, in meat, in fish, in milk.
Marty, if we can just squeeze in one additional. Lauren Erickson and then Rick Blumberg.
Yeah, Marty, great presentation. Really appreciate all the thought-provoking things that are coming from your initial presentation. Your tying of economic theory to infectious disease made me think about daycare and the economic phenomenon of more of our kids having their microbiomes affected through the mixing at a very young age with other children. And I'm wondering, does your group look at that phenomenon to see if there's differences? Because right now, as a father of children, now as a grandfather, what are going to be those potential downstream effects, not just to the use of antibiotics, et cetera, but of the way that we handle our children at these young ages? So
we know that daycare increases the transmission of pathogens. That's been shown with Shigella, with hepatitis A, Giardia. But it also may increase the transmission of commensals. And I don't think it's really been studied. We've begun to study that in mice through co-housing experiments. And I hope to be able to report next year about that.
Rick?
My kids have a loud voice, so. Marty, very interesting. And just to ask a question and perhaps propose an opportunity, the question is any ideas about the mechanisms by which H. pylori might confer protection from a disease in the lung that is asthma. And if one knew the molecular principles of that, another opportunity might be to reduce that to a pharmacologic preparation and then to be able to give that factor that is responsible for protection rather than having to infect with the organism. Could you comment on both of those?
Sure. So, again, I want to point you to the really wonderful work by Ann Mueller and colleagues at the University of Zurich. What we knew before Ann's work is that the T-cell components of the stomach in H. pylori positive and negative people are quite different. H. pylori positive individuals have a much bigger T-cell population, including much higher levels of T regulatory cells than H. pylori negatives. And there's some evidence from Karen Robinson that it's not just a localized effect, but there you can see increases in Tregs in the peripheral blood as well. Now, what Ann and colleagues have shown is that Helicobacter turns on dendritic cells in a way that makes them produce IL-18, which specifically recruits Tregs. And presumably, I would presume that that's the mechanism for protection, and that is that Helicobacter is involved in the setting of immunologic tone or immunologic thresholds, so that if you're not exposed to an allergen, it doesn't matter. But if you are, it determines whether you can turn off the immune response or not. And that's a very active area of research.
If you have time, Jesse Goodman had a question.
Well, it's just a really quick one, but the answer is probably 8 days long, which is, first that the risk of making all these associations, because antimicrobial therapy, at least in human populations, is also associated with all kinds of socioeconomic and other factors that could also predict obesity and things like that. But asking you, what is sort of the state of epidemiology or even any human clinical trials around this issue?
I don't know the answer, and we've been working with a birth cohort in England, the Allspox study, where 14,000 children were enrolled in the early 1990s. And the questionnaire data about early life antibiotic use is one question. Did you have antibiotics? But we showed that there was a significant association between antibiotic use in the first 6 months of life and measures of adiposity at age 7, at age 3.8 and 7. That was just published this summer in the International Journal of Obesity. From that same cohort, we now have evidence that the cesarean section increases risk of obesity, and that's been published from a group in Boston and a Canadian group as well. So we need large-scale epidemiologic studies through Kaiser and other groups that will really help us understand whether these observations and experimental models, these descriptions, are really have meaning. And finally, to answer your question, and it is an 8-minute answer, that's why we're working on mice, because we can control many of the important risk factors.
Marty, really appreciate all the time you've been able to provide and all the ideas and great sort of stimulation for what will be a lot of continuing discussion of the same over the next couple of days. We're looking forward to your return as soon as you can, as soon as the powers that be allow. And again, thank you very much